Good day, everyone. This is 2A Production, and I am Roy C. Borromeo Jr. from BSED 2A Science. So, here's the overview for the fertilization process. So, what is fertilization? Fertilization is a cell-to-cell recognition process that occurs between two distinct cells, a small asymmetric and motile sperm cell and large and non-motile egg cell. So the stages of fertilization can be divided into four processes, namely the first process is the sperm preparation, the second one is the sperm-to-egg recognition and binding, third one the sperm-to-egg fusion, and the last one is the fusion of sperm and egg pronuclei and activation of the zygote. The specific structures of the sperm and egg that are important for fertilization will be discussed, and experiments that led to the identification of the egg receptor for the sperm and the sperm receptor for the egg cell will be described. Membrane fusion of sperm and eggs is an incomplete understood process, but the discovery of proteins known as ADAM proteins on the sperm surface has suggested new mechanism to explain sperm-to-egg fusion. Finally, we will consider how fertilized eggs prevent additional sperm from fusing, or a condition known as polyspermy, and how fertilized egg is activated to begin development. So at the end of the video, you guys should be able to discuss the sequential nature of fertilization, in which order changes in the gametes or drive the process of fertilization toward completion. Second, explain the role of specialized sperm and egg surface structures in fertilization. You can describe how egg and sperm receptors were identified. You can also explain the current state of knowledge about sperm-to-egg membrane fusion and how sperm components are incorporated into the egg. And lastly, you can describe how polyspermy is prevented and the fertilized egg is activated for development. So before proceeding to the four stages of fertilization, here are some terminologies used in the fertilization process. So we have capacitation. It is the process by which the sperm becomes capable of fertilizing an egg. We have acrosome reaction, which is a regulated exocytotic event in which an apical vesicle in the sperm head fuses with the sperm plasma membrane. The acrosome reaction is triggered in response to egg factors. We have acrosine, which is a serine protease released during the acrosome reaction. We have cortical reaction, which is a regulated exocytosis in which apically localized vesicles or the cortical granules in the egg fuse with plasma membrane after fertilization. Next term is the zona pellucida, which is a coat surrounding the egg that contains 3 glycoproteins. Next is the galactosyl transferase, an oligosaccharide modifying enzyme that is usually found in the Golgi, but in sperm is on the cell surface. It is thought to be important as the sperm receptor for the egg. We have fertilin. It is an ADAM family protein on the sperm implicated in sperm-to-egg membrane fusion. It contains a fusion peptide resembling viral fusion peptides and a disintegrin domain involved in recognition. Next is the ADAM proteins. It is a family of proteins that contain a disintegrin and metalloprotease domains. So next is the pronuclei. It is the transitional male and female nuclei formed in the egg after fertilization. They fuse to form the diploid zygote nucleus. Next is the polyspermy. This is a condition in which more than one sperm fertilize an egg. Polyspermy leads to defective development. As I mentioned earlier, there are 4 stages to fertilization. The first one is preparation, in which capacitation and acrosomal reaction is in this stage. Acrosomal vesicle fusion is the membrane fusion event of this stage. The next stage is the binding. Species-specific interaction of gametes is in this stage. Third stage is the fusion. Merging of sperm and egg plasma membranes is the membrane fusion event of this stage. The last stage is the activation of the zygote, in which cortical reaction or the fusion of cortical vesicles with the egg plasma membrane and pronuclear fusion is in this stage. So, watch the video.
Good day everyone, this is Florence M. De La Paz and I'm going to discuss to you the gametes involved during fertilization. First, what is fertilization? Fertilization is the process by which the sperm cell that came from the male parent fuses the egg cell, the ovum, that came from the female parent to produce the specialized type of cell called zygote. There are 2 gametes involved, the egg cell and the sperm. The egg cell is the largest human cell. She measures 0.15 to 0.2 mm and just visible to the naked eye. The cell consists of a large amount of cytoplasm in which the nucleus is dissolved until just before the conception. Human eggs are arrested in metaphase of the second meiotic division and complete meiosis upon fertilization. Eggs are surrounded by a zona pellucida, which is a glycoprotein coat composed of 3 glycoproteins. The zona pellucida is not an osmotic barrier, however, it is a barrier to the sperm. This is clinically to assess the fertilizing capacity of the sperm. Sperm cells are the smallest human cell. They have 3 components. First is the tail. The tail contains the flagellar apparatus, which is composed of microtubules and accessory structures. The sliding of the microtubules is powered by the protein dynein. Next, we have the midpiece. It is located at the proximal portion of the tail. Midpiece contains a sheet of mitochondria produce the ATP necessary for the tail. And lastly, we have the head. The head contains the spermatic haploid nucleus. Overlaying the head is a membrane-bound vesicle, the acrosome. Acrosome is a lysosomal-like compartment derived from the Golgi.
Good day everyone from Group Fertilization. I am Regeline Enriquez of BSED 2A Science. First step, preparation of the sperm. Capacitation under this stage. Capacitation is the process by which the sperm becomes capable of fertilizing an egg. Acrosome reaction is regulated exocytotic event in which an apical vesicle in the sperm head fuses with the sperm plasma membrane. The acrosome reaction is triggered in response to egg factor. Ejaculated sperm is not ready to fertilize an egg when they enter the vagina. In response to the factor dilution of semen in the vagina, they undergo several changes which are collectively known as capacitation. The principal techniques of sperm preparation consist of migration, density gradient centrifugation, and fertilization techniques. For the migration method, it is based on the movement of the spermatozoa. For density gradient and filtration techniques, the method is based on the combination of the motility and the retention phase borders and adherence to filtration matrices, respectively. The semen consists of suspension of spermatozoa stored in the epididymis that, at the moment of the ejaculation, is mixed on the secretion of the accessory glands. These glands are mainly the prostate and the seminal vesicles, while the bulbourethral glands and the epididymis represent only minor contribution of the ejaculate.
Good day everyone. Today I will be discussing the step 2 of fertilization, which is sperm-egg binding. In humans, the process of sperm-egg binding is not so simple. The complicating factor is the thick zona pellucida, which keeps sperm from binding close to the egg plasma membrane. sperm receptor on egg. Dr. Paul Wasserman used a competition assay to isolate and identify the factor in the zona pellucida that was involved in sperm-egg binding. Dr. Wasserman incubated sperm with zona pellucida glycoproteins or ZPGPs he had isolated from unfertilized and fertilized eggs. He found that sperm pre-incubated with ZPGPs from unfertilized eggs were not built to fertilized eggs. Yet, when he pre-incubated sperm with ZPGPs isolated from fertilized eggs, which are known not to bind sperm, the sperm could still fertilize eggs. This showed that the isolated ZPGPs from unfertilized eggs contain a receptor for the sperm and that this receptor is modified after fertilization. In follow-up experiments, Dr. Wasserman purified ZPGP1, ZPGP2, and ZPGP3 and showed that only the CBGP3 could prevent sperm binding to eggs, showing that ZPGP3 is the sperm receptor. By treating ZPGP3 with agents that selectively hydrolyze protein or trypsin, nitrogen-linked glycoproteins or specific glycohydrase, and oxygen-linked glycoproteins or weak base, Dr. Wasserman showed that the part of ZPGP3 that was responsible for sperm binding was oxygen-linked oligosaccharide. Egg receptor on sperm. Dr. Barry Schur was studying a Golgi enzyme known as the galactosyl transferase. This enzyme catalyzes the addition of galactosyl residues from the donor nucleotide sugar, uridine diphosphate galactose, to the terminal end of oxygen-linked oligosaccharides. As in all enzymatic reactions, there are two stages in catalysis. The enzyme binds the substrates and the enzyme catalyzes the reaction and releases the products. It is important to understand that if one of the substrates is not present, the enzyme may be able to bind the available substrate, but will not be able to catalyze the reaction. This is important in sperm binding. Dr. Schur found that sperm, which have no Golgi apparatus, have galactosyl transferase on the surface of their plasma membrane. When sperm are ejaculated, they have oligosaccharides bound to galactosyl transferase. During capacitation, these coating glycoproteins are removed, allowing the galactosyl transferase to bind to other carbohydrates it may encounter, such as those attached to ZPGP3. The sperm that do encounter the egg and its zona pellucida bind CBGP3 through their galactosyl transferase. At this point, UDP galactose would normally bind to its site on galactosyl transferase. Galactose residue would be transferred into the oligosaccharide would be released. However, there is no high-energy UDP galactose in the extracellular fluid surrounding the egg, so catalysis does not occur and the sperm remains tightly bound to the egg zona pellucida. Acrosome reaction. As a result of a reverse binding of the sperm to the egg, the zone pellucida triggers the acrosome reaction. The outer plasma membrane of the acrosome fuses at the multiple sites in the plasma membrane and the contents of the acrosome are released. Two of the important components are acrosine, acerine protease, and nitrogen acetyl glucoaminidase. Acrosine bores a hole in the zona pellucida so that the sperm can reach the egg itself. Nitrogen acetyl glucoaminidase hydrolyze the oxygen-linked oligosaccharides in ZPGP3 to allow the sperm to detach. As a result of the membrane fusion, a new surface is exposed on the sperm and it is thought to contain the new binding sites for ZPGP3.
Good morning everyone, I am John Dave Duldulao, here to report the step 3, namely the sperm-egg fusion from the topic of fertilization. In contrast to cell division, which occurs commonly and frequently during the development of an organism, the fusion of sperm and egg is a rare event, restricted to a specific cell types. Sperm and egg fusion is a cell membrane fusion event essential for the propagation of sexually producing organism. In gamete fusion, as in other fusion events such as virus cell and intracellular vesicle fusion, membrane fusion is a two-step process. First is the attachment of two membranes through cell surface molecules, followed by physical merger of the plasma membrane lipids. Attachment proteins are responsible for the initial interaction between two membranes. In virus cell fusion, virus receptors interactions are mediated by carbohydrates and cell adhesion domains of proteins or other molecules in the plasma membrane. The similarities between cell-to-cell and viral cell attachment proteins probably stem from their common function and might provide predictive criteria with which to evaluate candidates as for sperm-egg attachment proteins to begin the fertilization process. The cumulus cell that surrounds an ovulated egg secrete a matrix containing hyaluronic acid. A sperm must first pass through this matrix and then interact with the egg's coat, the zona pellucida. This stimulates secretion of the contents of the sperm acrosome, an intracellular sac of molecules necessary for penetration of zona pellucida. Once through this coat, the sperm gains access to the perivitelline space and binds to the egg's plasma membrane. Sperm and egg membranes then fuse together.
Good day everyone, I am Marjules Domingo and I'm going to present the step 4 of the fertilization, the activation process or the egg's response. The immediate events after the fertilization includes the egg's effort to prevent the polyspermy. Polyspermy refers to the fertilization of an egg by more than one sperm, resulting in zygote to have a greater than a diploid amount of DNA. This causes in an early embryonic defect and arrest in development. Polyspermy is a great threat in fertilization and also the reason why there are multiple sperms into an egg to fertilize it and surrender the inviable zygote at the end of the development. After the sperm-egg fusion, the egg mounts the cortical reaction. The cortical reaction is a calcium-dependent exocytotic process in which the content of the secretory granules is released in the perivitellum space immediately after the fertilization, which serves to prevent the polyspermy fertilization. The cortical is propagated on the surface of the egg. In all eggs residing under the plasma membrane, there are membrane-bound vesicles known as the cortical granules. Cortical granules are specialized group of secretory vesicles that are randomly dispersed throughout the cytoplasm of an immature oocyte, then mediate toward the cortical cytoplasm during the meiotic maturation. The first enzyme is the ovoperoxidase. The ovoperoxidase in sea urchins catalyzes the cross-linking of the tyrosine residue in the extracellular matrix. This makes the extracellular matrix tough and soluble, and physical barrier was formed to prevent the other sperm from fertilizing the egg. In mammals, ovoperoxidase does not catalyze the cross-linking of the tyrosine residue to the point of insolubility. Also in mammals, its major effect is thought to be a spermicial agent. The second important enzyme is the hydrolase or the hydrolytic enzyme. The hydrolytic enzyme are bound in the protein matrix and in the course of the fertilization. Acrosomal hydrolases are releases in the acrosomal reaction and facilitates sperm penetration of zona surface. Acrosomal reaction. Acrosomal reaction allows the sperm by the exocytotic process of acrosomal enzyme to penetrate the zona pellucida of the surface. The species or the specific zona pellucida protein is a protein that serves as sperm receptor, also stimulate a series of events that leads to fusion between the plasma membrane and outer acrosomal membrane.